is Gustavo Stolovitzky, who is from IBM, who started the DREAM Challenges in 2006. And it's my pleasure to introduce him. He'll give the first talk. And then Steve Kornblau will follow him. We'll talk on the proteomics dataset, which is the focus of this challenge. And then I'll follow up with just describing a few innovations in the field. And then we'll break for a panel discussion where you're free to answer and ask any questions. And then finally, we'll finish off with a lunch. While Gustavo is setting up, I want to thank a few people who were absolutely instrumental in being involved in organizing the challenge. That is the group over here, my lab, all of them. I'll introduce some of them during the panel discussion. And also the people who hosted this conference, and that is people at the BRC, Lisa Ajita, who you saw walking back and forth, and Rami Swiden. In addition to them, we were sponsored by the Texas Medical Center and HATRIC, the Houston Area Translational Medical Research Consortium. So thank them.
Thank you, Amina. Thank you, everybody, for being here in this opportunity of telling you what we have been doing for this round of Dream 9. So I want to tell you a little bit the concept of crowdsourcing and trying to use community intelligence to solve biomedical problems, and then tell you a little bit more about Dream in particular. So first I want to introduce a few buzzwords that we all know what they have heard of them at least probably, but not necessarily know what they mean. So one of them is crowdsourcing, and crowdsourcing is basically obtaining services from typically an online community. And the use of it has been growing since 2005. In 2006 we launched Dream actually. This is in Google, this is in Google Scholar. This is in Google Scholar and this is in PubMed. So they mirror each other. But the notion is that there is more and more use of the concept that many people could have a contribution, even if you don't have this, the usual model of asking the best mind or asking the expert in the field. You ask many people who maybe are not necessarily the experts, but whose answers typically tend to be better than that of the expert. The second buzzword is big data, which is a strange thing that we call big data. Actually, I work for IBM, and part of our business is big data. But what do we mean by big data? Is basically the analysis and way to massage and transfer and process data that before we didn't have problems with doing that, but now we do have problems with doing that. We don't know how to analyze that because it's too big. It takes weeks to maybe extract some run, and then you have to do another run. So that's what we call big data. Big data is data for which we don't have solutions. And so if you put together the concept of big and complex data with the concept of crowdsourcing, and you try to tackle with those two things important scientific problems, what you have potentially is what we call a challenge, a scientific challenge. And to be more precise, what we call a scientific challenge in our case is basically the following. We have a cohort of subjects for which we know something. For example, as we will discuss later, whether if this cohort is affected with AML, some people might have had resistance to treatment, some people might have remission. Suppose that we know that, and also we take some data from this cohort in terms of, for example, proteomics, transcriptomics, clinical information, and so on. Now, if you are able to give this data, the usual model is we take data and we try to understand this. And so one group or research group tries to make sense of the phenotype given the genotype information. But what if instead of having this be done by one group, we have this done by many groups, let's say 50 or 100 groups, everybody thinking a little bit differently. So the idea is, can they, observing this, tell us the phenotype of the cohort that we started from, given that they don't know that phenotype. If they are, the few that are able to do this, we will have learned their method as a good method to extract the information that is valuable and probably go back and try to reverse engineer, what are the proteins that they have used to predict the phenotype and so on. And if that's the case, then in 3 months or 4 months, it is the time that the challenge lasts, we will have tried hundreds of methods, and that one group that could have done it very unlikely could have tried so many and found the right one for this problem. So what I'm trying to say here is that by doing a challenge, you can accelerate research, because really you can have a lot of people thinking about the same problem with the same data, and you can compare them. Also, we have, for the same token, democratized the data. So instead of having one group, massaging that data for, 2 years maybe, or 1 year and a half, and then trying to publish it and so on, and then, in the middle, the patients continue to have the problems, and we are not doing the research that they need and for which they have provided the data. However, if we give the data to the interested people to analyze it, we will have made that data have a lot more impact than otherwise. The third thing that we have done is evaluating objectively each one of the predictions of the people who had attempted to solve the problem, predict the phenotype from the genotype, because usually when one single group tries to do that, you use the same data in cross-validation and try, and finally there may be, even if you are very careful, some leakage from the data to your methods, and there is some problem with self-assessing, how well you have been doing. But what we have done is taken this out of the view of these method developers in such a way that they could have never used this information to make the predictions that they made. So the predictions are completely blind. Therefore, we have an objective evaluation, which is very important today because we know there have been a lot of problems with reproducibility of results. They are having problems with fraudulent attempts to claim panels of proteins or genes doing something that eventually they were proven that they didn't. And one thing that we are particularly interested in doing is to foster collaboration between a community to try to solve the problem, because we believe that there is what is called wisdom in the crowds. And I wanted to tell you about the story of wisdom of the crowds that starts more or less in 1907, when a Nature paper was published by this gentleman called Francis Galton, who's one of the founders of modern statistics. And Francis Galton, who at that time was 84 years old, he went to a county fair and saw that a lot of people, about 800 people, were trying to guess the weight of an ox. They were given values in pounds and submitting this to the organizers of this contest. And then the organizers, of course, measured the weight of the ox and found that the real weight was 1,198 pounds, when the average of all these people was 1,197, and closest to the closest expert trying to predict. So this is one of the examples, of the many examples, in which the collective aggregate of the guesses, or of the actual, not guesses, but noisy guesses, noisy predictions of different groups, balance each other, balance the overestimation and the underestimation, and what cancels out is the noise, but what remains is the signal. And let me tell you something that my son did a few weeks ago. He was given the opportunity to do a special project. So I told him about this wisdom of the crowds, and so he created this experiment. Here there are lots of glass beads in a jar, and he asked all his friends in his school, about 100 people, exactly, he counted 100 people that he asked, how many glass pieces there were in this jar. So here is the plot of the estimates. This is the estimated number of pieces from 100 to 700, and different people were up and down, right? But and when he told this to me, I couldn't believe it, because we analyzed the data and we found that the actual number was 307, and the average of it was 301, and you can see that only 2 people were closer to the real number than the average number. So now the fact that 2 people were closer means nothing, because if we had repeated this in some other time with the same people and with another set of another number, probably they wouldn't have been the ones that were closest to the real number, but the average would have been. So the average contains a robust estimate of the effect that we want to measure. With DREAM we have done a lot of this. Of course, it's a little bit more sophisticated than just averaging a number, because what we want to do is to understand whether people can classify or not, whether people can predict, for example, changes in phenotypes and so on. But the effect is the same, and invariably we have found that the wisdom of the crowd estimate, the aggregate, in whichever way you do it, there are different ways to do it, tends to be the best, second best, or third best. So the DREAM project this is last year's group. Amina, you should be here. I didn't find you here. Maybe you are around here. But we gathered together, and we every year, and we try to discuss the problems that we face. In principle, the kinds of things that we do are transcription and signaling networks, challenges on that, predictions of response to perturbations such as drug response to drugs, and translational research. And today we are celebrating our launch of the AML challenge. These are a few of the challenges that happened recently. This one, the HPN DREAM Breast Cancer Network Inference Challenge, was run last year. It's the one that Amina, Qutub, and her group won. And after that conference where everybody got together, Amina approached me and said, I may have an interesting data set that may be used for a challenge. And that triggered what you will learn about the challenge a little later. So finally, I want to say that today we are launching 3 challenges in DREAM. One is about predicting the change in cognitive, the decay in cognition of Alzheimer's disease patients based on some of their clinical characteristics and also their SNPs, the SNPs of these patients. We are also launching what we call a broad DREAM Gene Essentiality Prediction Challenge, in which we are trying to understand from a panel of about 200 cell lines what are the genes that are the most essential for the viability of those cells after following data of a project at the Broad Institute called the Achilles Project. But the most translational of our challenges is this one, the Acute Myeloid Leukemia Outcome Prediction Challenge, in which we really are trying to predict who in a population of patients, or whether one particular patient is going to be remitting or relapsing, and how long will it take, whether the patient will be resistant or not. And we hope that eventually that reaches the clinic and could be used in actual clinical care. And without anything else to say, I want to, is Steven? Yes. Steve did the magic that created the data for this challenge. So
this is Steve Kornblau from MD. Thank you.
So the next few minutes, let me introduce you to AML, Proteomics, our PPA, and better with this? Okay. And the data set for this challenge. So AML is a leukemia or malignancy of white blood cells. About 18,000 cases that will happen this year in the US, about 10,500 deaths, and overall about 25% are cured. At MD Anderson, we see about 1.5% to 2% of these cases each year in our single building. If you think about how many hospitals are across the US, we see way more than our share. Male predominance, whites and Hispanics get it more than African Americans or Asians. As you can see from the chart, your risk goes up a whole lot as your age goes up. Median age is 65 years. This shows how much or how little progress we've made over the decades from the 70s, 80s, 90s, and 2000s overall. And you can see that prognosis has improved more in the under 60 crowd than the over 60 crowd. But clearly there's substantial room for improvement. The traditional clinical prognostic factors for AML were age. If you were over 60, you did worse than if you were under. Cytogenetics, which were loosely grouped into favorable, intermediates, and unfavorables. Whether you had a prior hematologic problem, myeloid-dysplastic syndrome, maybe therapy related, you'd had prior cancer and had chemo or radiation therapy, and performance status. The problem with these, which I realized a long time ago, and many others as well, is that you can't treat these. You cannot make somebody younger, you can't reverse their history, and you can't improve their performance, make them healthier before you treat them. We tried that once, that didn't work. So you cannot therapeutically target these prognostic features. So you need prognostic features that you can target if you're going to improve outcome. So here's a newer scheme based on cytogenetics and some of the newer mutations that have come along. And again, you can stratify patients better, but the problem is that currently there are not any therapies that attack any of these particular mutations specifically. So you have information, but you still don't have a way to act on it. These are trials, phase 1 and phase 2 trials that we currently have open at Indiana and for leukemia patients. You can see that they cover a wide variety of different topics and potential methods or attack points within the cells. The problem with a lot of these is that they may work in small groups of patients. And if you think about the statistics, if you have a treatment that is gangbusters, but only 5% of patients, and you do a clinical phase 2 trial, which typically has 14 or 15 patients, the chances that you're going to have a patient who's going to respond to your agent is on less than 10%. And so there's a very high risk that you will discard effective drugs that are highly effective for a small group of patients because you can't figure out which patient should go on that therapy. So we need something that will allow us to do that. We've all probably read the very famous Hanahan and Weinberg paper on hallmarks of cancer. And all cancers supposedly have to meet these 6 criteria to become malignant. From the TCGA trial, or paper that was recently published, they identified 23 common mutations that are existing along with the limited number of fusion proteins that we have. And they were able to show that some of these correlate with or against others. Some will give you on circus plots stronger bands and weaker bands. But you can see that a lot of them are rather randomly associated. And this leads to a big dilemma. If you've got all these different mutations occurring in various combinations, how do they combine to meet those 6 hallmarks of cancer every time? Okay? And you really don't have a way to account for how they do that. And this is just mutations, but we know that there are other effects, microRNAs, the microenvironment, SNPs, that may affect this as well. So, not too difficult to take the leap of faith and say, well, proteins exist in networks and signaling pathways, and they translate the genetic event, on one hand, into what's going on in the cell, which has to meet those 6 hallmarks of cancer. And so it wasn't too big a idea to think that maybe there would be recurrent patterns of protein expression that, despite having multiple different upstream mutation patterns, these would coalesce into a more finite set of protein expression patterns, and that these would tell you which are the things to attack in a cell. And we hypothesized that we can use this to identify which therapy to match to which patient. So why couldn't we just use mRNA expression rates? Well, 2 reasons. One, the protein correlates very poorly with mRNA levels for the vast majority of proteins. But more importantly, we know that post-translational modification is crucial, and you can't measure phosphorylation, cleavage, uclination, assimilation, etc. on mRNA arrays. Could we just use traditional 2D gel electrophoresis mass spec? The advantage would be it's non-biased, you get a readout on all the proteins that are there. The problem was that the guys at Anderson that we talked to about this, and granted this was back in 2002, 2003, said that sure, they could do 2 patients a year. We had a lot more coming through. And they also needed an exorbitant amount of starting cells, which if we could take all the leukemia out of the patient, they could then study for us. But if we could do that, we wouldn't need it. So we realized that mass spec wouldn't work either, we would need a novel method. And around that time, Lance Liotta and Emil Petricoin had come up with this poorly named but useful methodology called reverse phase protein array, in which you print proteins on a slide, you probe it with an antibody. The disadvantage is that you have to choose the proteins that you're going to look for. It's biased. You have to have an idea of what you want to look for. And not every protein is going to have an antibody. So it was good for this, but it was good only for retrospective analysis. But it was very quantitative, bigger dynamic range than we saw with Western blot, good intra and intra-slide reproducibility. The way we do this, we print protein onto slides in 5 sphere resolutions, probe it with a highly validated primary antibody. We validated 280 of these so far, 230 are one use of this sample set. Probe it with a secondary antibody that has a signal. We precipitate a brown dye, scan the slide, use a program called MicroVGene or others available as well to convert this into numbers and do a lot of statistical manipulation to get rid of background. So on this data set, and we were fortunate to receive funding from the Leukemia and Lymphoma Society to do this, we created an array that had samples from 511 AML patients. 49 of these we had a paired relapse sample. Those are not part of this work. Samples could be either from the marrow or the blood, and we were fortunate enough to have same-day samples, 140 of them, and from these we learned that blood and marrow were equivalent for protein expression. Samples could have been prepared fresh on the day we got them, or from cryopreserved cells, and to see if there was a bias in using cryopreserved cells, we made samples from frozen cells from 58 where we had fresh, and for the most part in AML, fresh and frozen are indistinguishable. That's not the case for ALL. Our patients were typical of all AML patients, a little bit more male predominance, age around 65 years. We have a very high percentage of patients that had an antecedent hematologic disorder and prior malignancies. Consequently, we have a lower than typical percentage of patients with favorable cytogenetics and a higher percentage of patients with unfavorable cytogenetics. For this data set, we wanted to make it a little bit more homogenized to answer the questions that we have. And so some of these 511 patients were not treated at MD Anderson, so we eliminated them. And some of these patients died early, under 4 weeks. You could have all the good favorable prognostic features, but if you die of a heart attack on day 3, we can't really score you for outcome. So we cut those patients out. And then we limited it to a group of more standard therapies that use an agent called cytisinorabinuside, which is the backbone of most AML therapy. We cut out some of our more experimental trials. So this left us with a group of 323 patients. And they're pretty typical of leukemia patients. Roughly 2/3 achieve CR, 2/3 of those relapse. And at the end of 4 years, and we have 4-year follow-up on all but 2 or 3 patients, we're left with about a quarter of the population that's still alive. The data fields in the data set include traditional demographics, prior history questions. We have genetics, we have mutations on FLT3 and NPM1 and RAS, many clinical laboratory and flow cytometry measurements, therapy outcomes for whether patients achieved remission, relapsed, if they got a transplant, are they alive or dead, how long they lasted in each of those states. And then we have the proteomics in which we stained with 231 antibodies. So on this slide, white is a total antibody, yellow is a phosphoantibody, blue is a methylation, and the pinks are cleavage states. And you can see that we're very heavily weighted to signal transduction, autophagy, apoptosis, a lot of cell cycle regulation, a lot of other proteins that regulate other functions within the cell. We've used this to publish several papers on individual proteins while we've been working towards a way to have a more global omnibus analysis. And we've even recently, including a paper with Amina's group, looked at functionally related groups of proteins and the ability to build networks. But what we've really wanted to do was to look at proteins as function-related classes together and see what patterns we see. And this is work we've done with Amina's group and Wendy. I think Amina's probably going to tell us more about that in a second. But we're very excited to be able to look at networks, and we can break patients down by cytogenetics or by particular mutations and see different patterns of protein expression and activation within those, which we think will allow us to eventually target therapy to that. And I'll stop there.
I'm actually going to talk a little bit broader than my own lab's work and introduce you to where I think the field is innovating in looking at high-dimensional data. But I will show that a similar circus plot to what Steve showed there. I'm actually going to walk you back to when I started at Rice and met Steve. And this is in part to encourage you to, whatever domain you're coming from, to start thinking about competing in these types of challenges. My background was small scale. I was studying hypoxic response, how a cell responds to low oxygen, from a point of view of mathematical modeling. And what I mean by that is logic-based modeling, or ODEs, or PDE modeling. And I met Steve at a conference related to this work, hypoxic response, and he said he had this amazing data set of patients, and he was across the street over at MD Anderson. This was a conference in Colorado, by the way. And I said, sure, I'd be happy to take a look at that data set. It brought us into a very different domain. It's one of the most comprehensive data sets out there for leukemia patients. And it has dimensions which I had never seen before in the data that I was working with. And my lab is also house experimental, but again, we hadn't done these high throughput analyses. So that adventure brought us to where we are today and this dream challenge. So that pathway that I was studying from the point of view of more small-scale modeling fits in here in context. You can see hypoxic response pathways. This is a circus plot showing you how those proteins fit in with the larger scale sense of all the other functional groups related to AML that are a part of the data set that's being opening up for DREAM. So the question I have is, how do you act on this data? What do you do to make a decision point on whether a particular patient will have a particular outcome or not? How do you interpret that data? So that's where the field of high-dimensional data analysis is going. And I want to make an analogy to where I was actually as a graduate student. Before FitBeat was available, I was recording myself. I'm a scientist, I like to observe different things. So I was recording data points on myself during graduate school at Berkeley, over 200. Little things like what I ate, what I ran that day, whether I went to bed on time. And I was trying to look at modifying my behavior. And in this case, I thought I was addicted to licorice. You can pick your choice of how you would want to modify your behavior and what data points you want to record. But the question is, how do you then take these data points, and I looked back at what I had recorded, and I was successful in culling some of my addictions, like licorice, but others not. Where are we in understanding this type of high-dimensional data and acting on it? Well, we're actually quite good at taking these features and building different types of models. This would be another group of models which are looking at the individual data points, individual features, and how they map onto a decision. One is the set in blue, one is the set in orange. Two different sets of models, and some of you can tell right away some of these are more predictive behaviors than others. So if I zoom in and I look at the individual features as unique things that are predictive of whether I will be addicted to licorice, whether I'll eat licorice that day or not, you can see things like 6 hours of sleep or less and miles run would be predictive as correlative, as strongly predictive as whether I would eat licorice that day. So we have these models, but one question I have is if I want to make an action on this information, on these models, I need more than just that prediction of the point. I need to know my confidence in that point. So the new area of innovation I see is that when you look at those points, you're not just predicting the point, but you're predicting how stable your estimates are. And what is your confidence interval in that point? So if I actually want to make an action to stop my licorice addiction, I may not pick and think that sleep is anywhere correlative because, while it might be a predictor, there's a lot of noise associated with it. And the confidence intervals in that makes it a more risky choice in changing my behavior than how much I ran that day. So there's a lot of new algorithms that are coming up in looking at how do you predict your confidence in a point rather than just predicting that feature. And so that's an exciting area of innovation in high-dimensional data. Another one is actually one that's been catalyzed by the DREAM competition and much of the work by what Gustavo has been doing. And this is a very nice paper that he published a couple of years ago using different types of methods and figuring out signaling, which are the best at predicting particular signaling motifs, so biological signaling motifs. And some of them are better than others, and the ensemble is better than all of them individually. And this paper walks through exactly which features of which models are optimized for particular types of motifs, and how the ensemble is good at predicting the motifs together. So that's a new area where you can not only rank, but optimize ensemble approaches by the use of the crowd. Another area of innovation in high-dimensional data is looking at learning algorithms to mimic human discovery. So if I take an example that was actually an example that I saw recently at a presentation by Carrie Wagstaff at JPL in Caltech, where you give a group of people who want to be able to predict new life forms on a different planet characteristics of existing life forms and figure out what is unique and different about something that they hadn't thought about. Where could they find and identify new areas? So imagine this plot here that you're seeing here is a plot of the dimensions reduced for each of these features. So it could be principal component, your choice of dimensional reduction technique. Each of these features, each of these points here represent an animal. And you're the scientist looking at this, and you're asking to identify things you know. And once you identify what you know, you can then start to uncover what you don't know. So an analogy would be taking this data point on the far right here, and looking at popping up what that means. What is that feature? What is that animal that's predicted? And in this case, it might be, for instance, a reindeer. And you're saying, well, I know that sphere. I know those animals. So I don't need to study that area anymore. So you block off that area, and you pick something which is as further away as possible in your dimensional space to look at. And that other area you also might know. That might be a rhino, for instance. You start to iteratively do this to walk through your feature spaces until you reach something which you didn't know. So basically, you're taking the uninteresting data and allowing it to inform the very interesting parts, the new and innovative parts. What might be something which you could discover and hadn't looked at before. So there's another area of innovation. This is another one. Actually, learning algorithms have their history back into the 70s and when artificial intelligence started. But these are areas where I see new innovation in high-dimensional data analyses. The other one is addressing this problem. If you look at this is an Excel chart of actually Steve's RPPA data, and it's actually what he gave me when I first started with him. And this is what we were tasked with, understanding this data. But you can see how it would be very difficult to look at this data and make any type of interpretation of this data in its raw form. Well, there are new techniques to start to allow us to look at this data in its raw form. And this is partially what my lab won the DREAM competition for last year. And that's looking at this type of data in a circular heat map that's interactive. So you can start to see that MAP2K is highly expressed when STAT3 is lowly expressed in this area. And you can start to click on the points and interact with it. And that's another point that's new for this challenge, the AML Dream Challenge this year. And 2 students and I, working in the lab, have a startup company that's looking at this high-dimensional data. This is done a mock-up of what you're going to be seeing online and available to you that Wendy at Hugh and Alex Bisberg put together for you today. So if I'm going from this Excel data chart to something that is more interactive, you have what we're presenting now for the first time for the DREAM9 AML Challenge visualization. So what you're seeing here is a series of modules which display a data, protein heat maps, survival curves, and clinical correlate heat maps for the AML Challenge data. In the data selection panel, you can select proteins of interest. I've selected 3 here. Clinical correlates of interest, groups, and patients, and their corresponding levels are shown on the right in circular heat maps. You can see each spoke of the wheel here represents a single patient, And as you go along radially, you see the expression levels for these 3 proteins in the upper right-hand heat map for all the patients in your group, from high, which is bright red, or vice versa, from low, which is bright green, to high, which is bright red. Here I have grouped now and sorted by a particular protein. I can do the same for the 2 clinical correlates I have by just clicking on that particular clinical correlate's name, which I've done here for age at diagnosis. I can also look at groups. So if, say, I want to look at gender. Here, if I click on gender, I can look at female patients and male patients specifically. And here, dark green is for male, and yellow is for female, which pops up in the survival curve below the data selection panel in the survival curve area. If you want to specifically looking at one of those groups, say male gender, you click on the rim of the circular heat map, and it selects that group alone. And those particular protein expression levels for male gender and their clinical correlates show up. You can also look at other groupings and resort by clicking in the center of each button. So I also can look, for example, at prior malignancy. Here I show it in the clinical correlates and the protein expression levels, and I can deselect gender. Another nice feature of this visualization is to look at individual patients and their expression levels. So here I've clicked on one patient, a series of patients, and you can see that their protein heat map expression levels and clinical correlate heat maps show up and are synced with their survival curves, which is a red button that shows that patient's survival rate. So I encourage you to look at this visualization demo and play with the data this way, and also give us feedback on this new visualization tool. So with that, I'd like to conclude by thanking everybody who's been involved in this challenge. There's been so many people behind the scenes. One person who's not here who I'd like to acknowledge is Kelly Norrell, who's at IBM, who's really did a lot of the work in parallel with Dave Noren here in my lab on testing the data set. And everybody in my lab, they did a tremendous effort in organizing the competition, so all of them, I give them a hand. Obviously, Gustavo and Steve, who had this amazing data set which was accumulated over the course of over a decade. So I'd like to thank them all for their contributions to this challenge. And then thank you and welcome you to the panel discussion, where we'll have Gustavo and Steve come up to answer questions about the challenge, along with Wendy Hugh, Alex Bisberg, David Noren, and Byron Long in my lab. All 4 of them competed and won the sub-challenge last year in Dream 8, and have worked on different aspects of the challenge this year. Byron's been working on integrating with the Synapse Sage's interactive collaborative website. Dave Noren's been doing the testing. Wendy and Alex have been working on the visualization platform for you. So with that, I'd like to open it up to questions from the audience. Yes.
Hi, Stephen. Question about the RPPA data. So we know the cleanup data is available after supercurve and all that. Will the raw level data before supercurve, will that also be made available?
No. There would be, we can talk with you about it and we could provide that. It would be too massive an amount of data, I think, to give out the raw data for each slide, and people would need to have a proprietary program like Regine to be able to read the raw data from some of those files. So that would be a barrier, I think.
There's a question back here. Yeah.
So, as a layperson who has no real background in this and is not affiliated with any academic lab, is there any place for someone like me in this challenge? Like, there are like groups that say, hey, I want an extra body, or is this more intended for like different academic groups at MD Anderson or something like that?
I'll speak quickly and then I'll give it to Gustavo to answer. I would say it's open to everybody, and the benefit is having people from diverse backgrounds compete. And additionally, forums like this, and there's also going to be a later hackathon, it'll allow you to interact with people. We'll send out a list of people who are attending, if everybody agrees, so that the email is open to people involved, to match up with teams.
So what is your background?
I have a background in artificial intelligence and informatics.
So the It's an interesting question. Let me open the question a little bit more with respect to other trends in the area of challenges, which is the gamification of challenges. You take a problem which is well framed in a particular area, but you can translate it into something that could be operated as a game. Like, for example, we saw to understand the folds of RNA in a game called Eterna, or to try to help with protein folding in Foldit. So I think that in this particular challenge that we have, the AML challenge, we felt that there is so much domain expertise that could be leveraged that cannot be translated in that way, that is a little bit difficult to pose it in a way that is that you treat the numbers without knowing that they are genes or proteins and so on. So I guess there is a lot that will be lost. But I would be particularly interested in knowing whether, if you take the data as numbers that could be meaning anything without the need of knowing that they are genes or proteins or phenotypes or clinical covariates, I wonder whether you can win. Because in the old days of CASP, some of you may know critical assessment for protein structure prediction, which people were given the primary structure, the primary sequence of the protein, and they had to predict the three-dimensional structure. People who won were people who knew nothing about biochemistry in the beginning, and the physicists that supposedly know how things interact in space and have force fields and so on, they lost miserably in the first because they didn't have a good calibration of their force fields. So I think there is room for people like you to try, and it's an invitation to learn a little bit more about the disease and try to understand some of the domain information.
I think David.
Yeah, I just want to mention that I don't think it's been mentioned yet, but we have a wiki site, and we've done our best to make it very detailed, with just for people like you in mind, to really kind of level the playing field so that we can really bring expertise from all sorts of disciplines, to bear, their particular talents on this problem. And I think maybe we have the URL somewhere.
Yes, it's on the flyer that should be outside. Thanks, Dave.
Just two very simple questions. First, First, I didn't. Can you summarize the objective of the challenge again? My understanding was you were to find best prognostic factor which best predicting the outcome, right? That's the goal of the challenge. Second, how do you evaluate the results? Who's winning? I didn't get that from the presentation.
So I'll let Gustavo and Steve answer the first part, and then Byron has been working on the testing with Kelly at IBM.
So there are, I think, 3 clinical endpoints. Can you predict who's going to go into remission? Can you predict who's going to relapse? Can you predict how long people will survive?
Yes, and the remission duration is the second one.
So, but I think that the interesting thing that Steve has in mind when he is studying this data is also this, beyond the fact that you can predict the phenotype from the genotype distribution. Can you see what are the features that are interesting in order to determine, for example, potential targets for therapy? That is harder to score. We are not going to score. What we are going to score is whether people can predict correctly the phenotype. But hopefully, those people that can predict best the phenotype are also the ones who have found the features that are most meaningful. If you want to speak about scoring.
Sure. Initially, there will be released a training set of data for which you can create and calibrate your models. There will be weekly deadlines, and you'll be able to submit your new version of your results. And we will score those against a test set of data that we hold back. Each week, we will release the results and relative rankings of all the submissions, not only for the weekly period, but including all the previous periods also. So if you change your model, you'll be able to see if it was an improvement or not.
So those training based on some synthetic data, you have those training and
Real.
initial, those are all real data, but something you have answered previously from previous challenges.
You're given a training set which includes all of the real data and the outcomes, so relapse, remission, duration, survival. And from that, you're asked to build a model, which is best predictor, given features and not knowing outcome, what is best predictor of outcome. And so then you're scored on your predictive of data that's withheld, and that data is you're scored on whether you can predict outcome, the confidence that you could predict that outcome.
Okay, so on that note, I'd like to know, is the data sufficient, enough to evaluate, how good the algorithm is? Steve, you mentioned that initially you start with 511 patients, it's narrowed down to 323. So is that the total number of patients that available, and from the 323 you are split into the training and the validation, whatever.
So I'll give this to Dave Noren, who's been working rigorously since January on testing and figuring out whether we have a good training and test set.
So, yeah, the 323, I think the number I'm working with is more on the lines of 200 or 300. So I think we did a little additional filtering than Steve had mentioned. But what we've done is we've taken 2/3 of the data set, and we've tried to ensure that the distribution of the 4-day clinical covariates and the 231 antibody readouts or proteins follow similar distributions between the training and the test sets. Not sure if that, I think you're asking me whether or not it's predictable.
If there's enough power.
So we did some preliminary testing here at Rice alongside with Gustavo at IBM and some of his colleagues there. And we do show that there is a level of predictability, but we won't, we don't want to say that what we've found is the best that can be done. We definitely think there's a lot of room for improvement.
Let me add something to this before going to Pavel. Is that there is another aspect to your question, whether we have enough statistical power in the validation set to find the people who can predict without penalizing them because the distribution is broad, because there is not too many data points, and therefore the p-value might be only big if we ask a lot from people, to have to predict correlations very close to 1, even though that's not the case. I think that we don't have that problem. We have tested that you can, in many other challenges, that having 100 to 150 is enough to find people who have sufficiently good predictability, even if it is not really up there. There's a second point that I want to make with respect to your question, which has to do with the generic dream question that we ask ourselves. When do you know whether a problem is solvable? When do you know whether a problem is solvable or not? I believe that crowdsourcing, in a way, there is no proof of a negative, right? In order to prove something, you have to prove a positive. However, if you crowdsource a problem to 100 groups and none can get a statistical significant result, then you have a good indication that the data set with which you are trying to address the problem is probably insufficient. Now, if you have done it yourself, maybe 1, 2, 3 methods that you tried, it's hard to say. Maybe someone else has a better idea. But if nobody in the community, at least today, knows how to analyze this data to squeeze some information, then probably that means that we need to add something to the data set in order to answer that.
I have 2 questions. One, are we allowed to use external data in addition to what you provide? And second, can we use one of your supercomputers, IBM or Rice?
So yes, you can use any external, as long as it's not data or information, as long as it's not something from the data set. So if you've been exposed to the data set before, then you can't be a part of the challenge, but anything outside. And last year in Dream 8, that actually was the deciding factor between the winners of small challenge, and the winners of the synthetic challenge. They used external data and public databases.
And yes, you could use IBM supercomputing power, right? But you have to pay for it. No, you have to ask for it, and we'll try to provide it if you lack sufficient compute power. There's one more thing I wanted to say with respect to what other data you can use. That comes with respect to the gamification issue. That's another thing in which using all the existing information about AML could be useful if you go back and try to see whether your prediction somehow is related to some prior knowledge. I think this is necessary because we are trying to solve not a game like some gamification people are trying to do in order to reach more people, but actually a real problem. And for that, anything is game, except for going to one of the surviving patients and taking a sample from him or her. Yes.
So, and I guess I would like to really encourage people to use external data sources to better understand the proteins and how they interact with each other. From my perspective, the way that this will be most clinically useful is if we're able to tie back certain groups that predict or that how they do based on their proteins, because those are targetable. Again, I can't make a 65-year-old 52 so that they will do better, but if I can identify what the key protein is in that older group of patients, then maybe I will have one of that long list of drugs that are in development, and I can specifically go back and target it to those patients. So I would highly encourage people to use external knowledge about what the proteins are, who they interact with in formulating their patterns.
Yeah, I just wanted to speak to the visualization. It's actually another tool that can help you sort of understand what's going on with connections in the data. So as you saw, you can segment the heat map and just see how patients are actually surviving. So that allows you to check the test data set and see, maybe give you some clues or some ideas into where you might want to start.
While the visualizations are really cool, my question is mainly on the data sets. How do you put together your data sets for these dream challenges? Are they usually from a single institution, or are they collected from a number of different institutions?
This one is from a single institution.
What about for the other ones?
How do we put together the data sets?
Yes.
For example, the Alzheimer's disease challenge, we have three sources. One is ADNI, Alzheimer's Disease Neuroimaging Resource. It is public for everybody. So we cannot use that as a test set because that's public. You can go and get it. So that's our training set. But then we have two other groups that have contributed to specific test sets, and we had to coordinate that the covariates are the same so that we can predict and those are groups in Europe. There is another challenge that we have been doing, rheumatoid arthritis challenge, trying to predict the outcome of treatment on patients. And for that, we also use a lot of data sets, which implies a lot of data governance, a lot of MOUs, a lot of ways in which we make sure that we are not infringing any privacy issues, IRBs and so on. It can be done, it's a pain in the neck, and we have done it for several challenges, yes. But what is interesting also in the RA challenge, just one more thing, is that there was one consortium that said we want to produce a data set, basically measuring the SNPs, that could be used as test set, and that's what we are using as test set. So people can take some interest and say, I want to fund or I want to do the work for an extra validation set that will be used. And that has happened several times.
So, going back to if you're getting data from multiple places, and in some cases this actually even within a single institution, how do you normalize the data? Because people collect it with varying definitions, different machines have different calibrations and things like that. How do you normalize this? and thereby not getting into the same issue that I think you mentioned on one of your slides, which is, are we missing some of the potential drugs because we are discarding these drugs as not being useful in a certain population setting. If your data is kind of getting mixed up because of difficulties in normalization, it's not an easy thing. How do you overcome those kind of challenges?
Right. So let me take a stab at that, and maybe the rest of the panel can also contribute. Sometimes I hear that organizing a challenge seems to be easy, right? You get data, you make a website, and that's it, and then you score. It's much more than that. As you see, there was a huge effort in visualization in this challenge. There was a huge effort in dry runs to see whether we have signal. There were efforts in different challenges on batch correction, whether there is batch effects, whether for example, in the rheumatoid arthritis challenge, there are two different scoring ways of determining how well a patient is responding. One is the EULAR, one is the DAS28. So we needed to see whether there is correlation between those, because we want to use one for one purpose. What I'm trying to say is that we look at this and we agonize about this. And I have decided not to do challenges because the data was so noisy or so hard to do the batch corrections or the different artifacts that could come with the data. And we value, I see many of you hopefully will participate in this challenge. I feel responsible for your time. Because if you are working on it, even though you are learning something out of it, but you are given garbage data, then it's my fault. Because you don't have to know it. We take the data that you get. But we do agonize about it, and we try to make sure that there is very stringent quality controls on the data and batch effects. Anybody wants to add?
This particular data set has been rigorously tested, both statistically tested at MD Anderson and then tested and trained with both BioLab and IBM. And one more question, and then we'll break for lunch so everybody can have lunch.
Sorry, from lunch. So be very practical. Is that only the winners are allowed to publish papers with the data set? Or is that if we are not the top of the top selected, we can still publish our algorithms models with the data set by citing the competition and the institution?
So short answer is yes. We can publish on the data set.
Oh, yes.
On the algorithm.
Right, so yes, we want that to happen. We want people to use the data. But for that, first you have to ask Steven, because he's the owner of the data, whether he is satisfied with his own publication. After that, the data will be open for everybody to publish. We also want to publish the result of the paper, of the challenge, for which we ask for people to wait until they publish their methods, especially if they are going to use the result of the challenge as saying, we won or we got second. So we would like for them to wait a little bit until they publish that. Now, if it is just their method, without the data, of course, right? But, David told me something that has to be said. We will have had another guest here from Sage BioNetworks, but Stephen couldn't come and Thea couldn't come. So all this work we did also with a very important institution, a sister institution. Actually, we have partnered. I say that we married Sage BioNetworks. Dream married Sage BioNetworks. And so, in a way, what we are trying to do is to use their strength, especially their platform called Synapse, in order to run and score the challenges and make people collaborate through their collaborative platform. And so this wouldn't have been able also without the Sage BioNetwork folks. One more thing that I don't think that we mentioned. We had another partner, which is Kornblau, right? Why don't you mention that?
Yes, PLOS Computational Bio has already agreed to partner with this Dream Challenge so that we know we will have at least one paper that's collectively published in PLOS Computational Biology from the winners and people involved. So that's a really good, very positive response. And that was very quick. They got back to us within less than a day. And any other publishing partners, any other incentives will be listed on the website. So with that, I'd like to thank everybody up here, Gustavo, Steve, Dave, and welcome you to the lunch which is outside. Thank you all.
We would also like to take this opportunity to invite you to the Dream 9 Hackathon. This event will be hosted at the BioScience Research Collaborative Building at Rice University in Houston, Texas, and will be broadcasted over the internet. The hackathon is designed for participants to share their ideas and approaches in order to receive feedback from other contestants, form collaborations, and possibly join efforts in the Dream 9 Challenge. All hackathon participants will get a chance to present on their ideas on-site or over the internet during the Saturday morning session. The afternoon session will be reserved for networking and presentations on different software that could be useful in this challenge. During the Sunday morning session, participants will get a chance to again present on new developments and input received, while the afternoon session will be again reserved for other constructive presentations and networking. An abstract book will also be provided for all the participants. So please join us for the Dream 9 Challenge Hackathon on July 26 through 27 for valuable input and networking. Thank you.